Okay, well, good morning, everybody, and welcome to Grand Rounds in the time of the COVID pandemic, which means it's being done virtually. We're not out of the woods yet, so we're not, planning to reconvene in person, and I would suspect that we'll go through the summer in this fashion, and then make a determination when we resume in September as to how it's going to be done in the post-COVID environment, which hopefully will be substantially more normal. Before we begin, I just want to make a few announcements. Speaking of COVID, the pandemic continues, but locally it's moving in the right direction, albeit very gradually. It's the numbers of the infection rate are bouncing around, but the overall trend is down gradually. The hospitals, the New York Presbyterian Health System's inpatient census of COVID patients is now in the lower 600s. Just to give you a frame of reference, at its zenith in last spring, it was 2,000. And then it dropped to like barely 100 in the summer, and then over the fall and till now it's come up and then gone down. And we've had multiple sort of mini surges. So hopefully this gradual decline will continue to a minimal or zero rate. Vaccinations are still being administered, but our hospital and our medical school have really been among the leaders in the city and in the state in terms of getting the vaccine implemented efficiently and extensively. It's still a process that we're encouraging people who haven't yet or who were ambivalent, and also trying to extend it to the groups that were in the priority groups and also to patients. The variants have become an issue, but so far, no variant has proved to be resistant to it. There have been things that have complicated the process, such as the thromboembolic events that have occurred with the AstraZeneca Oxford vaccine in Europe, and now with the J&J. The J&J statistics have been really concerned, but not something that's spells the death knell of that vaccine. It's really a pause in the use of it till they figure it out. But it's 6 people, mostly young women, occurs early, meaning in the first 2 to 3 weeks of administration. And it's an embolic event as a result of thrombocytopenia. But hopefully this won't impede the use of that type of vaccine, which is the attenuated virus adenovirus form, as opposed to the mRNA form. So everybody get vaccinated, and we have to stay the course with respect to strict mask usage, social distancing, the same infection control. But even if you're vaccinated and even if you're immunized, in order to maintain optimal protection, and as we try and move back more towards on-site work and normalization. Also, just to mention, we next week will have the Glassman Memorial Plant Grand Rounds, and none other than our own Maria Okendo will be giving that lecture. Finally, we will also next week have a faculty meeting, which will be at 9 a.m. in the morning, but this is going to be a faculty meeting that's notable for 2 reasons. One is Interim Dean Rustige will be attending, at least for the beginning part, to make some comments. In addition, there's going to be some updates that are really important, so I'm going to put out announcements for everybody to encourage attendance. And then finally, also, there is this year, as part of our philanthropic fundraising efforts, the Gray Matters event, but like everything else, it's not the usual in-person one at the plaza that we've done in years past. This is going to be a kind of improvised virtual one. It'll be a panel discussion that'll be held April 22nd, 11 o'clock, 11 till 12 or 12:30, depending on how much questions there are. And the panelists will be 3 of our faculty: Jeremy Vanderweel, Brett Rutherford, Stephanie Lamel. And Linda Rosenberg and I will co-moderate that, but it'll be talking about the mental health effects of COVID and also what mental health care in their respective areas will be like in the post-COVID world. And lastly, I'll mention that tomorrow or Thursday, the New England Journal will publish its first article on a clinical trial with psychedelic drugs. It's a study by Robin Carhart Harris in depression with psilocybin. The study is a good study, but not a great study, but the journal feels that this is coming on so fast it needs to have some recognition of this emerging pharmaceutical area. Also, there was an editorial that will accompany it that I was asked to write, and it's trying to put this in some perspective and also provide some scientific cautionary comments. This will be followed within the ensuing 2 weeks by an article in Nature Medicine, first authored by Rick Doblin of MAPS, on the use of ecstasy to treat PTSD. So, the psychedelics are emerging and moving quickly, and this is kind of following in the path of ketamine and cannabis. And so it's something of great interest, but also something which is a double-edged sword if it's not handled properly. So it's something to pay attention to. Well, speaking of paying attention, our speaker today is none other than Stephen Kushner, a faculty member who's returned home, the prodigal son, so to speak. Stephen is a native of Philadelphia. He went to medical school at UCLA and did a PhD there with Alcino Silva, and then came to Columbia for his postgraduate training in psychiatry. And after distinguishing himself as an outstanding resident with academic aspirations and research interests, he took a road less traveled, and rather than do a fellowship at Columbia or some American university, he took an appointment at the Erasmus University in the Netherlands, and did extremely well because he really rose rapidly in terms of his academic status to the rank of professor, and was funded very generously by the European Union and the sources of sponsored research, with in Europe. Stephen is, and I was talking to him about this before, but in a good way, is somebody that I sort of think of as a academic polymath, because when you start out in an academic career and focusing on research, you need to really be very focused and disciplined in terms of not diffusing yourself. But Stephen's research really has taken him in a variety of different areas, disorders, life or age groups, and but in a very productive way that's been synergistic. His main focus would be called the area of genetics and functional genomics, but he truly is able to do this in a translational way, working in gene discovery in large founder populations or geographic isolates, putting genes into transgenic rodent models, but also doing phenotypic assessments of populations of interest that are related to the genes and the conditions that they confer susceptibility to. We were fortunate to be able to attract him back to Columbia, and it's now been, what, a year? Stephen?
Yeah.
You've been back a year, and
Yep.
he just happened to time his return with the arrival of COVID, so he hasn't been omnipresent for everybody to see, but he will be, and he'll share the work that he's been doing as he's been active during this period. I'm not going to bother to go through some of the accolades he has, the fact that he's received substantial extramural funding for his work. His publication record is quite impressive, but that he really is somebody who's, I think, credit to our training program and now our faculty by virtue of being able to be at the forefront of cutting edge disciplines and apply them in a very productive way to the study of mental disorders, and to do so in a way that's not just giving lip service to translational research, but is really reflecting it in the course of his laboratory. He's also co-director of our relatively newly configured or convened genetics interest group with Thomas Lehner, and has been active in trying to grow our genetics program in conjunction with the IGM that David Goldstein heads and the New York Genome Center that Tom Maniatis heads. So without further ado, I'm going to turn it over to Stephen and allow him to begin and also introduce his topic, which I think has to do with frontal striatal circuits and myelination. Is that correct?
Yep.
Okay, so we will virtually welcome Dr. Stephen Kushner.
Thank you very much, Jeff. I appreciate the introduction and the opportunity to talk to everyone. It's fantastic to be virtually back, but I look forward to, like all of you, when COVID allows us to move past this pandemic and be together again in person. Simon, is my screen projecting okay?
Yeah, we can see it fine.
Okay, great. Okay, I'd like to tell you about one of the lines of inquiry that we've been making that has sort of, started from the clinic and gone back, actually into the lab, and now we're moving back forward again. So I think this really highlights the kind of work that we're doing, and is a topic that is of great interest to us as well, because it's revealed a number of fundamental neurobiologic mechanisms. So I'm going to concern a lot of what I'm going to talk about today with schizophrenia and this disorder, and our attempts, like many of you, to uncover what some of the fundamental mechanisms that are going on. And I highlight these two as just examples of the enormous success that's taken place over the last years in elucidating the architecture of this disorder. So on the left is the PGC3 latest iteration of the GWAS, now with 270 genome-wide significant loci. And on the right is a paper that's just coming out, led by TJ Singh and the PGC, in which we're having the first opportunity to have really large-scale case control studies, in which we have now 10 and likely coming more as time and sample sizes get larger, of genome-wide significant genes that are really firmly implicated in the disease. So the opportunities for being able to understand the neurobiology of this disease is really starting to take off. The starting point for us, though, because we were in the era before we had the benefit of such genetics, was a very well replicated finding from the brain imaging literature that global white matter microstructure was abnormal in schizophrenia. And this is one of the most well replicated findings consistently over groups, not with the sensitivity and specificity that would allow diagnosis of individual people, but at the group level, very, very consistently found. And this example is from a large DTI study from the working group of Enigma, published a few years ago, but this is extremely well established. The question, though, is whether or not the white matter changes that are being observed in brain imaging, are they causal for the disease? Or might they be a consequence or epiphenomenon of the disease, a consequence of medication, a consequence of the disease progression, or do they have some etiologic role? And so the work that I'm going to show you was done by three great people in the lab: Christian Baukamp, Femke de Vrij, and Guy Spock. And I'd like to share with you the work that they've done. So this story starts with a rare variant discovery using a bit of a different approach than I referred to before, than the Schema Consortium used, in which there was large-scale case control studies. Here, we're doing a number of studies using family-based, more classical family-based analyses. So the discovery family that I'm going to tell you about comes from the Netherlands, comes from this so-called Bible Belt region, The modal number of children in the Netherlands is 2, which isn't generally for discovery analyses very amenable to such analyses. But this was a more unique family that had quite a high loading for schizophrenia. And so here you see on the right, this branch that's numbered, was the core nuclear family that we worked with. The proband was the father. He had schizophrenia, and his brother and sister, as well as his mother, all had diagnoses of schizophrenia with very extensive histories of hospitalization. His brother, unfortunately, committed suicide, and he and his wife, they had 9 children, and 4 out of the 9 children had diagnoses of schizophrenia, it was a quite compelling family that we met. You'll note that that there are no children of this 3rd generation. They are now well into their 50s, and that was actually because, just as an aside, a relatively common phenomenon that we see when there's a large expansion in terms of the number of children and affecteds. In this case, this family was not very biologically oriented. They're not a family that you sit down and they start talking about genetics, but instinctually they understood that there was something very heritable that was likely going on, and made a pact. Actually, the children, in this 3rd generation, made a pact that they would not have children because they were concerned about transmitting a potential, a genetic risk to a next generation. Long story short, I'm happy to get into the details if anyone wants to ask, but essentially what we did was classic linkage analysis and looking under, the maximum LOD score peaks, and combining that with exome sequencing to look for co-segregation between the disease and either missense or severe loss of function mutations in the exome. And what we found was a single variant in which there was 100% co-segregation between the disease and a predicted damaging missense mutation. This was at the alanine 131 to threonine position in the CSPG4 gene. And you can see here that this mutation is carried exclusively in the affected individuals from which we have samples, and in none of the unaffected individuals. Of note, and I'll come back to this, this protein has 3 laminin G domains. These are extracellular matrix domains, and this A131T mutation falls in one of these laminin G domains. This protein structure was available to us that's modeled after the Norexin extracellular domain that has a similar laminin G domain. And in silico, there's a prediction for misfolding as a consequence of this alanine to threonine mutation. In order to try to further examine this association and validate this, we turned to an independent study that had been done in families of Mexican and Central American ancestry that had been done a few years back through the NIMH. And the leading candidate region that was identified through the linkage analysis was on chromosome 17 at Q21, but there was a second suggestive linkage peak that exactly overlied the CSPG4 gene. And so we were able to request those samples and resequence them throughout the CSPG4 gene, and we found a second variant that segregated to the extent that we had samples between affected and unaffected, and gave us a second opportunity to be able to look at this association. We were able at the time to look within the so-called Sweden schizophrenia exome study that was a predecessor to the schema study that I referred to earlier, in which there was nominally significant case control enrichment for this particular variant, and the Dutch variant that I showed you before was absent entirely from cases and controls. Interestingly, this second mutation is in a third laminin G domain, also predicted to change folding perhaps in a similar way. A bit more unstructured this domain is, but nevertheless, a clear effect on the folding of this domain as well. Now, in order to be able to look at the biology here, we turn to the advances in induced pluripotent stem cell reprogramming. And so what we did was we obtained iPSCs from 3 affected and 3 unaffected siblings in order, as much as possible, to be able to standardize genetic background. We spent quite some time actually trying to look into making isogenic controls. The major problem is that the CSPG4 gene has 11 pseudogenes, of which multiple of them have highly homologous sequence, not only in the gene overall, but in particular in the area around the mutation that exists in this family, really making the design of guide RNAs, or we even looked into zinc finger nucleases for TALEN-mediated editing, very, very difficult. And so we opted for this sibling-based control approach as an alternative strategy. We've spent quite some time, it's published, so I won't go into it, but a protocol in order to be able to differentiate neurons in 2 dimensions, as well as astrocytes that derive from a common neural progenitor cell. And we do that through embryoid bodies, and that allows us frontal patterned neural progenitor cells that have the capability of being able to be expanded and freeze thawed in order to preserve the genomic integrity and the passage number of the iPS cells that we're working with, and be able to reduce the heterogeneity of batch effects by being able to have as a starting point a cell type that's further along in differentiation. And so, as I said, we're working with forebrain-patterned neural progenitor cells, and we're able to see when we do this differentiation, the classic patterns, of the various cerebral cortex layers with all of the canonical markers that you would hope and expect, as well as, evidence morphologically of synapse formation. And so one of the first questions that we wanted to ask really is, the leading hypothesis in the literature has to do with synaptic dysfunction. And, this is where the convergence of the genetics up till the present has appeared to converge. And we wanted to ask, do we see evidence of this? Do we see evidence in our cells of neuronal or synaptic dysfunction? And so, long story short, 2 different postdocs spent a long time, doing extensive amounts of characterization of electrophysiologic properties at the single neuron level, at the network level, synaptic activity and excitability recordings, action potential characteristics, and essentially there's no consistent finding at all that we found. There's nothing that's robust that was standing up on replication. The electrophysiologic properties that we saw were largely unchanged. And so this really led us back to ask, then what might be going on here? And, over the time of the course of the study, there's increasing amounts of work that's being done in, single cell RNA sequencing, allowing us a better and better understanding of the cell type expression of CSPG4. And what we came to understand, through the emerging data was that, CSPG4 is a really restricted marker, specifically of oligodendrocyte progenitor cells in the brain. And even within the oligodendrocyte lineage, this is hardly expressed in more mature oligodendrocyte, myelinating oligodendrocytes, and really restricted to those obligate progenitor cells that are the precursors, the obligate precursors of myelinating oligodendrocytes. And so we went back to our iPS cells, and we, instead of differentiating them along a neuronal and astrocytic lineage, we differentiated those neural progenitor cells into oligodendrocyte progenitor cells and the oligodendrocyte lineage. And the very first observation that we made was when we just attempted to do a basic characterization of the expression of the CSPG4 protein within these cells. And what we saw very clearly was that there was a highly abnormal subcellular distribution of CSPG4, or what it's very commonly called NG2, the protein, a very highly abnormal subcellular localization of the NG2 protein. And in particular, we saw high co-localization between NG2 and calreticulin as a marker of intracellular retention within the endoplasmic reticulum. And if you remember back, and we'll get into this more, but if you remember back the in silico prediction of the folding abnormality, that would be highly consistent with such an abnormal subcellular localization. What you're seeing here are each of the individual lines. So I'm showing you the full distribution of all of the data, because each of the dots corresponds to each of those 3 patient and 3 control lines. And the errors are the errors on the distribution for experiments that were done for each of those lines individually. So this was a quite robust effect that we saw. The second aspect that we saw very clearly was a drastic change in the morphology of those cells. So you can see that in these very representative images here, and you can see this, rightward shift, on this cumulative distribution plot of area, in which the patient cells are much smaller and have, a much reduced cross-sectional surface area than the control cells. Second is their cell viability. Just keeping these cells in culture, we saw that spontaneously the patient cells were having great difficulty remaining viable and remaining alive. And it was very hard actually to keep them in culture compared to the controls, which behaved very consistently with our experiences with other iPS-derived OPCs. Now, as a consequence of the lack of isogenic lines, because of the pseudogenes that I referred to before, we did a series of complementation analyses to establish the mechanism genetically by which the phenotype was emerging. So within the pedigree, you could see that there's a dominant, autosomal dominant pattern of disease inheritance. And the question was, by which mechanism might this mutation be acting? Is it acting through loss of function or gain of function? And so we did a series of complementation analyses by transfecting the control OPCs with mutant CSPG4. And what we found was a recapitulation of the phenotypes that we had seen before in the primary lines through these complementation analyses, now both with the A131T mutation, that original Dutch mutation, and in addition now with the mutation that was found in the Latino population, in those of Mexican and Central American descent, recapitulating the abnormal intracellular colocalization, as well as recapitulating the decrease in survival of those OPCs. In addition, there's just as an interesting side note, the valine to glycine at 901 mutation. This caused a series of intracellular inclusions that we believe are colocalizing to endosomes that were highly, highly abnormal. We did not see that phenotype in the A131T mutation, but this is something that we're continuing to follow up to understand. But suffice it to say, in both cases, there really seems to be a dominant acting effect that we think is essentially a gain of function in terms of the deleterious impact of this putatively misfolded CSPG4 in the setting of these mutations. Now, as I said before, these OPCs are the obligate precursor to myelinating oligodendrocytes. And so in addition to a basic characterization of the OPCs and their viability, we also wanted to look at the extent to which there was an ability for these OPCs to differentiate, perhaps if we can get to the point of moving past the differentiation barrier, to what extent do we see intact or not myelination. And so this next part of the work was done a combination of by Femke and a very talented student in the lab, Mark van der Kroeg. And essentially what they had been developing in an effort to get more and more robust and realistic models for IPS differentiation for modeling neuropsychiatric disorders, is to develop an adherent organoid protocol in which we can make significant progress in trying to standardize what currently is a very challenging heterogeneity for the cerebral organoids that are most widely used in free-floating conditions, in which there's great heterogeneity, unfortunately, in the size and distribution of those cells. Our attempt here was to try to develop a protocol that was much more standardized. And what we've now developed in a 384-well format that's very screenable, that's amenable to high throughput kinds of analyses, in wells that have X and Y dimension of 1.8 by 1.8 millimeters. What you're seeing here is an overview of one entire well using this adherent organoid protocol that we've developed. And that has allowed us, and you can see this has a very nice cortical layering. So, here represented by the upper layer marker CUX1 and the deeper layer marker CUX2, we have very nice, layering between upper and deeper layer cortical neurons, as well. You can see here the separation border between them in higher resolution. And in addition, we really see the emergence of spines in a way that is not typically observed in other monolayer IPS neuronal culture, in which we really see the formation of mushroom morphology type spines, more classic spine formation that's consistent with the kinds of synaptic activity that we see. We have very robust synaptic formation with co-localization between all the markers that you would like to see between synapsin and PSD95, for example. And the density of those synapses is much higher, again, than what we would see in 2D situation. I won't belabor this, but we're able to do very nicely calcium imaging and look at the activity and synchronicity of these cultures and demonstrate that they're dependent on synaptic activity, as well as all the classic complement of channels in the neurons. There's radial glia and astrocytes emerge from here, and very nicely also the distribution of morphologies of astrocytes that's seen in vivo as well. Microglia we've been able to add, and that's a topic that we're getting increasingly interested in, to be able to supplement these with microglia, because of course they're coming from a different germ cell layer, and we're able to look at both activated and quiescent microglia in these situations. Now, we're able to do the experiment that we really wanted to do in this system by looking at oligodendrocyte precursor cells and myelinating oligodendrocytes in this system from patients and controls. And we're able to implement a myelination assay in which we're able to look at the expression of MBP and labeling along axons, and essentially, just to boil it all down, but I'll come back to this and we can discuss it more in the question and answer, is that the bottom line is that there's a gross decrease in the ability for these OPCs to be able to differentiate when they carry the A131 mutation into myelinating oligodendrocytes. And we see a very significant deficit in the MBP surface area and in the axonal myelination in these organoid settings. And lastly, and really this is just anecdotal, but I'd like to show it because it was an interesting and important moment. I think we went back to the family, and there are so few family members that we couldn't do the classic kind of brain-wide analyses. So we really held off on doing any kind of brain imaging until we might have a hypothesis. And when we had these findings in cells, we came back and asked, okay, to what extent do we see, really brain-wide global abnormalities in white matter microstructure? Now, I referred to before that, that is canonically what you see at the group level, in schizophrenia, but to what extent is that phenotype manifest and also identifiable with, at the individual level. And what we found was, in both of the affected individuals that we were able to scan, was a substantial departure outside of the distribution, the normal distribution. Here is the full distribution of 294 matched controls. And in both of the patients who are carrying this mutation, significant abnormality in global white matter microstructure.
Steve,
Yeah.
the potholes are hyperintensities?
So they're not hyperintensities. What they are is a slightly different definition. So the fractional anisotropy is defined at the global level, across the whole brain, and you get a single value there. The potholes are an attempt to deal with the fact that from individual to individual, you have small areas of contiguous voxels that fall below a fractional anisotropy threshold. And they seem to be distributed in a stochastic way from patient to patient. So there have been a lot of attempts to try and assign them to specific tracts, but that hasn't been that strong. And so there's a metric that came up in which you do kind of the stupid thing of just counting up the number of these regions of contiguous voxels that fall below a fractional anisotropy threshold. And then the question is, for any given person, what's your burden of the number of these regions distributed across the brain? And so, you can see here on the y-axis that, in a controlled distribution, a distribution, of people who is without mental illness, on average, they're having about 3 of these. And, these are age matched. So these are people in their 50s. And then, in the patients, from the family, we're seeing 10 and more potholes, of the like, and you can see that distribution. Those 2 individuals there, their contiguous voxels are represented here.
So the potholes is basically is focal FA anomalies.
Okay, so for interim summary, we're nominating CSPG4, as a novel candidate gene, for familial schizophrenia. We believe that, additional validation would be really important, in order to be able to definitively establish this. But on the basis of, the evidence that we have so far, we think that it's a gene that is worthwhile to pay attention to. We think that as a consequence, OPCs are a cell type that warrants further investigation, for schizophrenia pathophysiology. And I want to make an important point here. The earlier results that I showed you before suggesting, the absence of neuronal and synaptic dysfunction. First of all, those are in cultures that are lacking OPCs or oligodendrocyte lineage cells. And second of all, we're not at all saying that, the underlying mechanism of schizophrenia in these individuals does not involve neuronal and synaptic dysfunction. I mean, I would concede, of course it does. What the issue is whether that is the primary etiologic cell type. And what we're arguing is that in this particular case, we think that we might have a family in which the primary etiologic cell type may be in the oligodendrocyte lineage, and in particular oligodendrocyte progenitor cells, and that the presumably resulting neuronal or synaptic dysfunction is likely a second order effect of that primary etiological abnormality, in the non-neuronal glial cell type.
How are you able to distinguish etiologic from pathophysiologic, though?
Well, we think that we are able to do that in this case simply because of the exquisite cell type specific expression of the gene that's mutated. And so this is a rare situation in which we think that we can, make an assertion that it's unlikely that neurons are the primary affected cell type simply because they don't express this gene.
But isn't in rare mutations, the functional groups that those are accrue to are those? I mean, why would those be not meet the criteria that you're using for your oligodendrocyte cell type mutation?
Well, they could. So I think in cases in which we have more restricted cell type expression, that is a real opportunity to be able to ask, how the genetics data informs us about cell type. And they're starting to be, I left it out, but we could discuss it, afterwards. There's starting to be some, real sophisticated, interesting opportunities to leverage the genetic data, and the cell type distribution of the expression of those genes to try to elucidate how do we get from, the genetic findings down to cell type? How do we take that next step? And certainly, for schizophrenia, the overwhelming, major cell type, are neurons. That's not a big surprise, but that is, really, a major finding. But that doesn't mean necessarily that in all cases, that is the primary etiologic cell type. And again, we think that this is, one situation in which there may be a non-neuronal cell type that is the primary etiologic one.
And not to belabor it too much, but then would you say the same about interneurons or not necessarily?
In principle, there could be such a gene, such genes that could be interneuron specific that could allow us to draw that conclusion in the cell type analyses that have been done so far on the basis of the schizophrenia GWAS data in particular. There is some real enrichment, strong enrichment in interneurons, although glutamatergic pyramidal neurons in the cortex, for cortical neurons have continued to come out stronger in terms of their association, but there does seem to be an interneuron signal, and actually, I'm in the time I have remaining, that's exactly how I'm going to transition. That for interneurons, there is a compelling, set of data there. Okay, so actually, Jeff's comments, nicely make a transition. So, question is, how might the deficits in myelination give rise to schizophrenia? And that's what I'd like to concern myself for, for the remainder of the talk. The work I'm going to show for really talented people in the lab: Jeffrey Stadehouder, Demi Brzee, Maria Pascual Garcia, and Sarah Hijazi. So this starts back from work that was first published from David Lewis's lab, in which there is a deficit of parvalbumin expression that has gone through quite a circuitous route in asking questions about whether there's a deficit in gene expression versus a deficit in that cell type. I think this is largely a settled issue in terms of the gene expression abnormality, and couple that together with the quite well tread road in terms of the gamma deficit in schizophrenia during working memory tasks, and lastly, of course, the elegant findings showing the amount of control that PV interneurons have over gamma, and linking together the possibility that at the functional level there's something doing with these PV interneurons in schizophrenia. That's a big hypothesis that's out there. And we've got this tremendous cell type, neuronal cell type diversity in the cerebral cortex. This is of course what we were discussing just a moment ago, and I'd like to specifically focus on two subtypes that are arising from the MGE, together, at similar times during development, the parvalbumin, fast spiking interneurons, and these SST somatostatin interneurons. So, question is, might there be a pathophysiologic convergence between these PV interneurons and the findings there in postmortem brain and functionally, and these abnormalities, that we're seeing, in myelination at the group level, and then together with, the findings that, that we had from this family-based study. And so, if we go back a few years, our first question when we were thinking about this was, well, are these PV interneurons even myelinated? And our informal survey of, a variety of, neuroscientists was, why would these cells be myelinated? Myelination is for aiding conduction velocity, and these are really local interneurons. So how can you get much benefit out of axonal myelination in these locally projecting interneurons? But the graduate student very wisely did not pay attention to, the assumptions that a lot of people were making. and decided to actually specifically ask this question just to make sure, whether or not these cells were myelinated. And, long story short, in terms of the workflow, and I'm happy to discuss this more if people want details, but basically we're making whole cell recordings. In this particular case, we're, in the medial prefrontal. We fill the cells, we do recordings, we fill the cells, we reconstruct them, we stain for MVP, and then we ask whether or not the internodes that are there, to what extent and where might they lie along the axons of these various neurons. Okay, so first result is that in 3 different brain areas, we see very clearly a myelination of the PV positive, these parvalbumin fast spiking interneuron axons, and there's differences in the proportion of MVP, the proportion of internodes that are arising from this cell population that changes across the various layers in these different regions and is different across the region. So a few highlights. For example, in layer 2-3 of S1, of primary somatosensory cortex, 40% of the MVP in that area, 40% of the internodes come from PV interneurons. So quite an overrepresentation compared to the proportion of neurons that are there. In CA1, in stratum pyramidale and stratum radiatum of CA1, up to 80% of all the MVP and all the internodes that are there are coming specifically from PV interneurons. Interestingly, the Schaefer collateral pathway, the pyramidal neurons are specifically not myelinated. So then we asked at the single cell level, so how is myelination distributed across PV cells? And long story short there, we did extensive number of reconstructions, and we were able to find back internodes on 218 out of 222 of the PV cells that we did reconstructions. The other 4, is it possible that we just missed them or they actually unmyelinated? Regardless, minimally a very high proportion of PV interneurons are myelinated in the neocortex. We also extended this to human brain. We have a really nice collaboration with the neurosurgical department in which, in the rare cases where there's a deep tumor that needs to be resected and they have to pass through relatively healthy cortex in order to get there, instead of discarding the dissected tissue, we were able to accept that and then be able to use a very similar workflow as we did in mice to be able to fill these cells, record from them, reconstruct their axons, and then do post hoc histology to ask how myelin is distributed along these human PV interneurons. And what we found, this is such a human PV interneuron from the superior temporal gyrus, in which you see very extensive myelination, actually much more so than we see in mouse even. Here's an example, of more than 1 millimeter continuously of myelination of a human PV interneuron in temporal cortex. Here's an example. Here's another example, and you see, the topographic classical distribution, which is very similar in mouse and in human, in which there's a biased proximal topography of the distribution of those interneurons on the axon, and also with discontinuities at the branch points. That's also very classic in this, in the PV cells that we've made these reconstructions. Here's another one, again from this one, also from superior temporal gyrus, very extensive myelination. We don't see myelination this extensive in mouse. So one of the first questions that we wanted to ask, for myelination of pyramidal cells in the cortex, there's been shown to be an activity dependence. The suggestion is that somehow the activity of neurons is somehow signaling to the myelinating oligodendrocytes to signal the need, for myelination or for restructuring or reorganizing the myelin distribution. And so we wanted to ask to what extent that might hold true also for PV interneurons. So we used, again, a similar workflow, but now we're using DREADS to be able to drive chronically the activity of these PV interneurons, we used a 2-week, twice daily CNO injection. We drove these cells pretty well, pretty hard in some ways, but we wanted to really put ourselves in position to be able to ask the extent to which there's an activity dependence to the myelination of these cells. And the first thing that we saw very clearly is that, in the cells that are being driven, that there is an increase in internode number that's specifically at the sort of middle level branch order, around, somewhere between branch order 5 and 8 is where we really see the biggest shift in the distribution. There's maybe a small effect on internode length. In this analysis, it wasn't quite significant. Maybe it's there or not, but at best, it's a relatively small effect. The much bigger effect is the increase in the number of internodes. But what we didn't expect, and we found, and these are adult mice. These are mice, that are 8 to 10 weeks of age. We saw a substantial growth of the axon when we were driving these. So this is, really a finding of adult morphological plasticity of the axon of these PV interneurons in response to activity. And then when we looked, what we found was that, yes, although the number of internodes per axon increased, the proportion of the axon that's myelinated has not changed. So the mechanism by which there's an increase in myelination as a consequence of activity seems to be driven by a morphological plasticity. And then secondarily, there's an increase in the number of internodes, which is proportional to the percentage myelination that axon had before the activity was increased. So we then wanted to go on and ask, can we identify, can we answer 2 of the major questions? One is why the GABAergic interneuron myelination disproportionately involves PV cells. So I went through this very quickly before, but if we look at the various cell types, the various GABAergic cell types, PV cells explain the vast majority of all of the interneuron myelination that's seen from GAD2 expressing cells. And so the question is, what is it about these PV cells that they are disproportionately the GABAergic interneurons that are being myelinated? And second, can we say something about why there is this proximally biased topography of the internodes that I referenced before and showed you, in terms of why the internodes are primarily on the proximal part of the axon? So we use a very similar workflow as I showed you before, but now we add structured illumination microscopy imaging in order to make more detailed measurements of the axons to try and get at this question. And I'm going to skip a little bit through this very quickly, but basically, we used the SIM imaging to be able to compute the axonal diameter all across, the axon, the reconstructed axon, in order to be able to get those metrics more locally. And we validated this through a number of different ways. We were able to look at the empasamputans and how that overlapped with the myelination. And second of all, we were able to use a virus expressing APEX2 in order to do electron microscopy, in which the APEX2 enzyme allows us to do DAB staining that is EM visible. And so what we found basically is that if you plot, so here's where I'm going to show you a number of these plots in the subsequent slides. So here's how this plot is built. It has 3 metrics on it. One, so each of the dots is a different segment along the axon. And the operational definition of a segment is the uninterrupted interval between 2 branch points. So each dot is a different segment in an axon, and then putting together all of the various cells that we've reconstructed. So the segments are labeled either white or green, color-coded here, for whether or not that segment is myelinated or unmyelinated. And then on the y-axis is the diameter of the axon in that segment, the average axonal diameter in that segment represented by the dot. And on the x-axis is the length of that segment from branch point to branch point. And what you can see is that the myelinated segments are very highly predictive by the bivariate model, in which you have a threshold, a minimum threshold for axonal diameter and a minimum threshold for the segment length, for the interbranch distance. And when both of those, and only when both of those are met, those criteria, then you have a myelinated segment. And so let me show you now, first of all, the results of the EM. So that's very consistent with our finding of axonal diameter, and I'll show you much more data in this regard, but roughly at 350 nanometers is the minimum threshold for myelination. And so here is a reconstruction, series of reconstructions, and the data resulting from PV cells in the neocortex, in which you can see that the threshold for axonal diameter is again around 350 nanometers, and for the interbranch distance, that threshold is around 18 micron as the minimum interbranch distance, below which, even if you're super threshold for axonal diameter, you have a very low probability of being myelinated. And similarly, if you're super threshold for interbranch distance, but the diameter is too thin, you also don't have myelination. So it really requires the bivariate. And if you do an ROC analysis on this bivariate, you see very high, area under the curve and very high accuracy of prediction. Whereas if you look at just the two univariate models, they're much less predictive. The diameter tends to do much better than interbranch distance alone, but both of them are inferior to the bivariate. Now, we wanted to test the causality of that, and so we had, in the lab, in the neighboring lab, mice that had a floxed TSC gene, this gene that sits in the middle of the mTOR pathway, and one of the many things that that pathway is doing is controlling cell size. And we knew from all the wonderful TSC work that has gone on that knockout of TSC changes cell size, and in particular that loss of function mutations of TSC increase cell size. And so that gave us the opportunity to ask, what if we change the morphology of these cells? Do we change their myelination? Particularly if we just change that specifically in the PV interneurons. And so we did that. We also, in parallel, did a similar approach with UB3A, the causal etiologic gene for Angelman syndrome, in which it had been previously shown that, in the Angelman syndrome model mice, that you have smaller cell size through as yet not worked out mechanism, but nevertheless a way for us to be able to regulate cell size. And what we saw was that the manipulations were the way that we had hoped. These are all specifically cell type specific deletions just in the PV cells, and we see an increase in soma area in the TSC and a decrease in soma area in the UB3A mice, and we see a similar shift in the distribution of the axon onset diameter, as we would expect. And so what the finding is, if we do these reconstructions and make these plots, is we are shifting the distribution of morphology in the way that you would expect, but notably the thresholds, and these thresholds are determined independently for each of these, the thresholds stay very, very stable, suggesting that the biophysical rules that underlie myelination in the brain are very much determined on the basis of the morphology of the axon, and that's highly predictive of whether or not a given segment locally on the axon will be myelinated, regardless of how you're shifting the morphology of that axon. And so then we wanted to ask, well, if we go back to these somatostatin cells that I referred to earlier, these somatostatin interneurons that are not myelinated, they emerge similarly from the MGE. They're developing, with a relatively similar neurodevelopmental course, but they are not myelinated. Is that determined by their axonal morphology? Or might there be some other kinds of mechanism? But So if we reconstruct those cells and make the similar plots, again, what we see is hardly any myelination. In the 10 reconstructed SST cells, we found one and only one internode, on one axon that, interestingly, happened to exceed the minimum thresholds bivariately. So again, adhering to those rules, but these are generally unmyelinated cells. Now, if axonal morphology is the critical determinant for why PV and SST cells are differentially myelinated, then what if we just make the SST cells bigger? Should that be sufficient to induce their myelination? And so we made a cell type specific deletion of TSC to enlarge the SST cells. The soma area is larger, the axon onset diameter is larger, just as we saw before. And now, lo and behold, we start to see myelination. And if we do the reconstructions, in the wild type cells, out of 26 SST cells that we reconstructed nearly the full axon, we found at least one internode. And I think on one of these cells, there were 2 internodes, and on the other 3, there was 1 and only 1. Let's say roughly, on the order of 15% of the cells, but over 90% of the cells that had, enlarged, cell size, we see myelination. And again, this enlargement is cell type specific. And so, here's what now the curve looks like. Again, the critical thresholds remain the same, but you have a shift in the morphology of the axon, and consequently, you have just many more segments that exceed this bivariate threshold and result in myelination. And again, from the RSC curves, you can see that the bivariate analysis is superior to the univariate. Lastly, we wanted to ask, to what extent, are these axonal morphological determinants present in human neocortex? And we turn back to the similar workflow that we looked at before, in which we reconstruct and make these curves in human, and we see very similar thresholds in human as we see in mouse. The RSC curves maybe are not quite as good, but this is also with the caveat that this is being done in human neocortex from post-surgical tissue, but the qualitatively the RSC is very, very similar, and the distribution is very similar to what we see in mouse. Okay, so in summary, we find a potential etiologic contribution of oligodendrocyte lineage dysfunction to schizophrenia pathophysiology. We've really contributed to understanding this previously unappreciated myelination of fast spiking parvalbumin interneurons. And we think that is a locus of potential pathophysiologic convergence that warrants further study. The GABAergic interneuron myelination, I think, is strongly determined by axonal morphology. That seems to be true in mouse, where we can really do the causal experiment, and by correlation and association that seems to hold also in human neocortex. And we also think that rare variant functional biology and family-based studies, are really obviously making increasingly important contributions to neuropsychiatric disease pathophysiology. Okay, we're just very briefly, just to show you something that's very exciting now, we're continuing with these family-based analyses, and as Jeff alluded to, we're moving more toward, work in more genetically unique populations. So in particular, in areas of the Middle East and villages and regions in which there's high rates of consanguinity, and and instead of looking at dominant inheritance, now looking at recessive modes of inheritance, where we've ascertained a quite large population of such families to be able to look at potentially recessive mutation. And here's an example in which we have a genome-wide significant linkage peak on chromosome 9, that exceeds the classic maximum Lodge score. The variant, is, it's absent, in the public databases. It's predicted as damaging. And if you look at the RNA sequencing data, now again very enriched in the oligodendrocyte lineage, in the gene in particular in this family, but not anymore in the oligodendrocyte progenitor cells, but now in the myelinating oligodendrocytes. And so we're really interested, and enthusiastic to follow this up and continue to pursue this question of the extent to which there might be some important pathophysiology that's being contributed through myelination in the human brain for, the etiology of schizophrenia, at least in some of these, families that we're identifying. Okay, I'll stop there and glad to take questions. I want to thank all our funders and, the people in the lab, and really looking forward to, getting all this up and running, here locally, at Columbia and New York. Thank you, everyone, for your attention, and I'll be glad to take questions.
Thanks, Steve, for really description of a lot of very elegant work. I have kind of a couple of zingers for you, but before I, launch them, I'm going to ask some of the more specific questions from the attendees. So one question is, do oligodendrocytes have a lifespan, or does the brain continue to manufacture defective cells?
Yeah, thanks, Ralph. This is a great question. Actually, Jonas Friesen, has done some beautiful work using carbon dating from, a nuclear accident that happened in Scandinavia that allows the ability to be able to look back at this and ask, the lifespan of some of these cells. And it turns out that oligodendrocytes, neurons are the longest lasting cell type that we've got. Aside from this, these local niches where there's questions about adult neurogenesis, and I don't want to get into that, but I think there is strong consensus that for the vast majority of the brain, there's no neuronal turnover. And so those are the longest lasting cells. But myelinating oligodendrocytes actually, are not far behind. They're very, very long-lasting cell type. There's remodeling of those cells, but they're very long-lasting cell type.
Another question is from Yun Wang. Latest research has shown that diffusion MRI, I guess they're referring to diffusion tensor MRI, does not specifically measure myelin properties. Do you think other types of acquisition sequences for myelin might be more informative?
Yeah, so I would love, and we've been working, together with, some people to do that. I've talked to Scott Small about this actually, whether we can really get at this, in some of the great cameras that they've got, at ZMBBI, these, very high Tesla, imaging devices that they've got, downtown now to do animal imaging, but try to find ways to be able to do in vivo imaging that's, much more specific to myelin. So, I mean, from my understanding, it's just to be, specific, at least from my perspective, it's not, the issue is that the DTI does not definitively measure myelin, but that whatever DTI is picking up, if there's an abnormality in myelin, you would see it there. It's just that there's other ways to affect the DTI measures and the fractional anisotropy that could arise from other biological alterations than just myelin. For example, changes in neuron packing or axonal properties could change as well. So I agree with you that we need to be very careful in making a one-to-one correspondence between DTI and myelin. But on the other hand, if there is an abnormality, in the oligodendrocyte lineage, it's, we should still see it in the DTI signal. And, that's the perspective that, this limited DTI imaging that we did before, is coming from, and, also, from the Enigma consortium. But I agree, we really need more specific. And to take it a step further, as I think as most people here know as well, the DTI imaging classically is really in these white matter tracts. But for example, all the interneuron myelination that I'm talking to you about, this is all happening in gray matter. So what we really would love to have is, metrics definitively of myelin that, are measurable out in gray matter, where there's hardly anything really that's been going on. And, to my knowledge, very, very limited, if any, data from schizophrenia about gray matter myelination, which is where we think that, some of the significant pathophysiology that we think we're tapping into might be happening. So if anyone has ideas about that, I would love to talk about that.
I'm going to just jump the queue a little bit because Gaurav Patel has two questions that pertain to the one you were just responding to. Have you looked at cortical myelination in humans? There are MR methods for directly assessing cortical myelination. And then secondly, cortical myelination density varies across the cortex. Do you expect a global decrease in cortical, or are you going to see potholes like you were measuring in with FA?
Yeah, so the first question is, the answer to the first question is no, we haven't. And I would really like to, I'd be happy to discuss offline about this. I think, there's really an opportunity, I think, to do this in schizophrenia in particular, to ask the extent to which the DTI findings, really extend to cortical myelination, and just to map that in general in humans seems, important neurodevelopmentally as well. What would be our prediction, in terms of in terms of the gradient of density of cortical myelination in this family? I'm not sure. Where I can tell you is, besides across brain region, as I showed you before in our analyses, the contribution of interneuron myelination across cell layer, between moving from deeper to upper layer neurons, there is a very clear gradient in both human and mouse. And so there, we would really expect to see differences that we would think will scale with interneuron density. But there certainly could be alterations as well in pyramidal cell myelination, which we're now starting to look at and ask to what extent they adhere to the similar threshold rules that that interneurons do. So far, it does seem like that is the case, but we don't have a definitive enough data set to be able to say that. But that would be my prediction, that I think we'd be more confident to make predictions across cell layer than necessarily across different cortical brain regions. Yeah.
Just Garo adds, we do have some in vivo measures to assess T1, T2 ratio and T1 relaxation. So Eleanor Simpson says, compliments you on the cell biology, and then asks, are there notable similarities in clinical phenotypes observed in the patients with the two different CSPG4 mutations, like age of onset, comorbidities, et cetera?
Yeah, thanks, Eleanor. This is a great question that we wish we could be able to answer. The problem is that because, in the valine 901G mutation, those were samples that had been collected, quite before, and that study was concluded, and there was no way in the consent for us to be able to recontact those individuals. We actually wanted to see, could we expand the pedigrees as well and do deeper phenotyping, but that just was not possible. We are looking for other ways to identify people with, who are carrying, both of these mutations, to be able to do more extensive phenotyping. But for right now, we're not able to answer, unfortunately, whether there's segregating phenotypes between those two mutations.
And a related question is, how typical is the symptom presentation of these families?
Yeah, that's a great question. So thank you for asking that, actually. I would contend that it would be very, very difficult, for, the great clinicians who, are here in our department. It would be very difficult for people to pick out, the people from this family who have schizophrenia versus, classical community schizophrenia. There were no atypical symptoms. They didn't have neurological abnormalities that stood out. This was not schizophrenia plus. It was really the only difference in this family, which I do want to point out. It actually goes maybe to Eleanor's question a little bit, is the age of onset was a bit late. So two of the individuals had their onset in their mid to late 20s. The other two in their early 20s. So this was shifted late. Now, we believe that that's likely an ascertainment bias. And so I would caution, we've tried to caution ourselves, and, I would say it as an extension in thinking about it, about, how much to read into that. And we think it's an ascertainment bias because if you think about it, if the father had very early onset severe schizophrenia, the likelihood that he would end up with 9 children, is lower than if he had later onset. And we already know from the epidemiologic studies that the fecundity rates in general in schizophrenia are quite, decreased compared to the general population. And so we think that, we have to be careful about reading into that because it may simply be an ascertainment bias that these large families do tend to happen perhaps in situations in which you have a later onset.
Do the families display the gender difference that's typically seen with female later onset?
So I don't think that we have enough data yet, to be able to say that. In this particular family, only males were affected, because it was autosomal, and these are not imprinted loci. There's no reason.
The other thing for later onset is in underdeveloped or developing societies, the age of onset is frequently later just because of the detection sensitivity. So, I don't know how you probably retrospectively were dating their onset. So it could simply be a kind of sensitivity or time from onset of symptoms to diagnosis that accounts for it. You see the same thing in Asian samples that we've worked with in interior China. Myrna asks, what role do you see PRS analysis playing in informing the results?
Yeah, Myrna, this is a great question. As we're getting more and more information about rare variants, the potential interaction with PRS and, how those relative loads interact is a really interesting question. So far, what's emerged is that, for the more severe rare variants, the higher risk rare variants, that you tend to see a lower PRS than typical schizophrenia, which is consistent with those rare variants being, mediated by a rare variant modality. But at the same time, in a number of family-based studies in which there looks like there's a Mendelian pattern of inheritance, you're seeing excess of, rare variant, excess of PRS burden in those families, suggesting that, polygenic risk can play an important role in familial transmission. I think the more family-based analyses that we do, probably will emerge, a similar story as, what we see at the individual level, which is a subset of these families are going to be determined by, actually essentially inheritance of polygenic risk. That of course will, to some extent dilute out over time, although in founder populations and isolates, that might be a different situation, but in general, that's the case. And then, you'll have a subset of families in which, have rare variants that are, inheriting, in a true Mendelian way. But the interaction, with rare and common variation is a fascinating area that I think the field's going to learn more and more about.
So Jeremy Vanderwill also is really complimentary on the work, and then ask, seeing your DTI potholes and this unusual family makes them wonder whether outlier individuals for this imaging phenotype are seen in the general schizophrenia population, beyond just mean differences. Rare genetic approach isn't often paralleled in imaging or other phenotypic analyses, but might outlier analyses be used in these contexts?
Yeah, so this is a great question, Jeremy. So yes, such outliers are out there, and it would be very interesting to do a similar approach where taking a phenotype, I think you're suggesting, could we take a phenotype first approach and then ask, if we, let's say, reprogram cells from those individuals, would it be, possible, to observe, would you expect to observe, a recapitulation of, abnormalities, for instance, in the oligodendrocyte lineage in those cases. I'm motivated by, having seen a few, well done papers, in autism that have done that. One of them that I'm thinking of, took a phenotype first approach. I believe it was for macrocephaly. This was a few years back from a group from Toronto. And so it seems like, this is a viable, possibility. It's a little more challenging because, of course, there's lots of potential roads to Rome. Why, there might be, this increase in, in potholes, for various people. Could there be different pathophysiologies, if you will, that lead to the same phenotype, which might be difficult to recapitulate for one particular cell type. But I think that it's a great idea for, for approaching the problem from a different direction to uncover the biology. I fully agree.
So in the remaining time, I'm going to just ask my question. So, white matter has not been a traditional area of pathophysiology that's been demonstrated in postmortem studies, and to the extent that it has in vivo studies, it's been mainly using methods that the physiologic and clinical significance of are unclear, the tractography or FA and so forth. So, I mean, if you were studying, leukodystrophies or MS or some illnesses that had traditional symptoms or manifestations of white matter pathology, so I'm curious as to, what sort of drove you in this. Was it an opportunistic thing that you found this gene that was for oligodendrocytes that led you off in this direction? And all the work that you've done on the cell biology, which is extremely elegant, in the mapping the diameter and length as the criteria which elicits myelination. But to what end? And do you think that this is going to be an avenue of research apart from what its basic science significance is going to be informative for schizophrenia or maybe other genetic developmental diseases?
Yeah, so, well, I'll say a couple of things about it. One, I think it's an open question. We have been, part of our motivation has been continuing to uncover the fundamental biology. And so, that in and of itself, has been very valuable. But, I would contend along the lines of the discussion we were having before that, we don't yet really know, the extent to which, this is, an important pathophysiologic component of schizophrenia or other, neurodevelopmental disorders, because, the imaging, has not yet, the imaging studies have not yet really been done. From the postmortem perspective, it's tough. I've had really good conversations with Andrew Dwork and other people about how to approach this from a postmortem, and it's non-trivial. So again, we were able to do the experiments that we were because we were able to get living tissue that we could fill and reconstruct these cells and do these kind of detailed analyses. It turns out just technically to be very difficult to be able to make these kinds of measurements in postmortem tissue. But there are some ideas that we're working on to try to be able to do that, but it's technically very difficult. The last is, the more, if you watch the literature in the last years of myelination, there's starting to be more and more impressive findings of how plastic myelin is, over the life course, certainly over the course of neurodevelopment, and how, myelin might be one of the unique areas of biology where there's some real distinct differences. For example, between humans and, for instance, the rodent models that we have. One of the things just qualitatively, I showed and highlighted, which is, how much more extensively myelinated these PV cells are, in human than, they even are in mouse. And so we think that the jury's still out about the extent to which, this may be very important for pathophysiology, and we wouldn't necessarily count it out simply because, there hasn't yet been, evidence pointing in that direction, from the prior postmortem studies that have been done. We're interested to follow the genetic studies. There have been some indications of enrichment in the oligodendrocyte, lineage cells, not a huge signal, but again, that may, be relevant, as etiology only, in a subset, of the cases. And again, because of the biophysical interaction, we think that, myelination may not just be determined by oligodendrocyte lineage cells, but as I showed, by axonal morphology. So it's possible that some of the pathophysiology does indeed involve myelin, but that may be as a consequence of alterations in the morphology of neurons as well. So we think that this is really worthwhile to continue to pursue.
I hope so. I mean, there is a certain complementarity that, the findings that you've demonstrated with your iPSC and organoid studies, in that even though cell number of different cell types in postmortem tissue in schizophrenia has not been found to be diminished relative to controls, the morphology of the cell, the length of the dendrites, the synaptic dendritic spine density is diminished. So, that's, and then also, myelination occurs maturationally, through adolescence, which corresponds to the onset of illness, which of course has been highlighted in the synaptic elimination hypothesis of Feidberg et al., and then strengthened with the complement gene gain of function finding from Carolyn and Stevens. So, there may be a kind of a generalized phenomenon that affects both, and the neuronal or the gray matter components of it, with Pat Goldman and so forth, have been just the most studied, and the white matter dimension of it, less studied until you stumbled upon it.
Yeah, and to follow the this logic that you're applying now, Jeff, also, just to make the point, myelination and the maturation of PV interneurons are, one of the last, major neurobiological, maturing features, again, consistent with the ontology, of the symptomatology of schizophrenia. And in particular, the frontal and prefrontal lobes are, the last maturing areas, and also the last places, where myelination, is complete. So phenomenologically, in terms of the developmental time course, it fits, that's all circumstantial, but, if it would turn out to be, an important part of the biology, that would help explain potentially, that those, longstanding clinical observations.
Well, I mean, schizophrenia is frequently characterized as a connectopathy, and why should it be limited to the synapse? So, in any event, great presentation. We'll look forward to seeing sort of the next installments as your work progresses, and good to have you here.
Thanks a lot.
Okay, so thanks everybody, and we'll see you next week, and for the faculty meeting and then the grand rounds. And in the meantime, stay safe. Bye-bye.